This is first page, right? Ready? Slow it down.
Are you curious if natural sweeteners are actually better than artificial sweeteners for inhibiting bacteria? I grew up with a grandma who believed xylitol, which is a natural sweetener, inhibits bacteria growth and tooth decay. She believed in it so heavily, she invented a candy made out of xylitol called Ice Chips Candy that is known to be beneficial for teeth. This led me to the idea to test if this is true. My hypothesis states that natural sweeteners other than cane sugar will inhibit bacteria growth. Yeah, okay.
Are you sure? I swear I did.
It will show that slide. Just do it. The reason cane sugar is the only natural sweetener that will not inhibit bacteria growth is because in my research, I found that it only helps with making preservatives last longer. It does not make things safe, it is not a substitute for proper acidity, and does not inhibit growth of botulism. Therefore, it does not inhibit growth of other bacteria. Okay. As far as the rest of the natural sweeteners go, there was evidence that each of them would inhibit bacteria growth. For the artificial sweeteners, my research led me to think that none of the ones I used 2 types of bacteria to inoculate my agar plates, E. coli and Micrococcus luteus. The reason I use these specific ones is because E. coli is gram-negative and rod-shaped, and Micrococcus luteus is gram-positive and sphere-shaped. Oh god, I feel like I'm still where I was at.
How did you read that?
I just knew that it was there.
This one should take a while.
Remember, this one has to stop. The difference is the outer casing of the bacteria. A gram-positive bacteria will have a thick layer of peptidoglycan, a sugar protein shell that the stain can penetrate. A gram-negative bacteria has an outer membrane covering a thin layer of peptidoglycan on the outside. The outer membrane prevents the initial stain from penetrating. During this lab, I put 2 grams of xylitol, stevia, cane sugar, honey, aspartame, saccharide, sucralose, and corn syrup into nutrient agar plates and then inoculated with E. coli and Micrococcus for 24 hours in a 37 degrees Celsius incubator. Then I counted the amount of colonies per plate to see which sweetener inhibited the most bacteria growth. After doing the lab, my hypothesis was shown to be incorrect. I discovered that most natural sweeteners couldn't inhibit all bacteria, while most people think they do. Some artificial and natural sweeteners can inhibit bacteria growth. The natural sweeteners, xylitol and stevia, were able to inhibit most of the bacteria growth for the Micrococcus bacteria. They both had about 200 to 440 colonies. All 4 natural sweeteners were not able to inhibit the E. coli. They all had thick lawns. Out of all 4 artificial sweeteners, saccharide was able to inhibit the most bacteria growth of the Micrococcus luteus bacteria. Only about 100 to 200 colonies grew. For the E. coli bacteria, none of the artificial sweeteners were able to inhibit the growth of bacteria. They all had thick lawns of growth. This makes sense that none of the 8 sweeteners could inhibit growth of the bacteria E. coli, as it is gram-negative bacteria that has a thicker cell wall made up of 2 walls of cellulose. I would like to fuck you right now. For further study, I would use more varieties of bacteria to see whether the sweetener is beneficial for inhibiting bacteria growth or not. Testing only 1 gram-positive and negative doesn't give accurate results whether the sweeteners can inhibit all bacteria growth. There are a variety of bacteria. I would like to test the bacteria for gingivitis because it is inflammation of the gums, gingiva. It commonly occurs because of films of bacteria that accumulate on the teeth, plaque. Bacteria involved in the etiology of gingivitis include specific species of Streptococcus, Fusobacterium, Actinomyces, Veillonella,